vector. I think your program it says the vector of Rosette. What that term means is a vector is something that transmits something somewhere else. For example, the southern house mosquito is a vector of West Nile virus. So this is this little mite is a vector in the same way that that mosquitoes vector disease. So there's actually a superfamily of mites called Eriophyidea. These are kind of strange creatures. They're four-legged mites. Now we all learned that spiders and mites have 8 legs. Well, these mites have 4 legs. They're really a different type of mite. They live typically inside plant tissue. There's over 1,200 different types of these Eriophyid mites. These are the only mites that are known to transmit viruses. And so when this Rosette disease was first discovered, it was immediately theorized that perhaps it was this little mite that was the vector. And sure enough, some studies confirmed that these were the mites that were transmitting the virus. Now these mites, this superfamily has got different sorts of names depending on the different symptoms that the mite species themselves produce. Sometimes they're called gall mites. You might have heard the term rust mite, blister mite, or just an Eriophyid mite. They're all referring to mites in this particular superfamily. For those of you who keep an eye out on turfgrass issues, we have an Eriophyid mite that attacks Bermudagrass. This is the Eriophyes cyndonis. And the picture on the left there, the sprig on the left, has that same witch's broom type appearance that you see with Rosette disease. And if you, in my experience, these mites are actually a little easier to find. They're a little more common because when you peel back the leaf sheaths on those witch's brooms, you can see these little tiny whitish, yellowish mites down underneath the leaf sheaths. So this is what you would see with a hand lens. Another Eriophyid mite that causes some common problems here in the Metroplex, the ash flower gall. This is Eriophyes fraxiniflora. And you may have seen these dark gall-like things on ash flowering parts. And these galls are not harmful to the tree, but they do persist long past the growing season, and so they kind of look ugly when the leaves fall off and you see these galls up in the tree. Some years we have a lot of those in ash trees. Some of these mites make these nail galls or finger galls, and you'll see these little bumps on leaves, and inside you'll have these little hair-like structures sometimes, and the mites will actually be inside these galls, supported by the hairs inside the gall. So it's actually, I guess, a benefit for the mites to have these galls formed on the plants. Those of you who hate poison ivy, you may have seen some of the galls like this on poison ivy out in the woods, and this is another Eriophyes mite. And sometimes the galls formed by these mites form little open galls or blister-type galls, as in these two pictures. And again, the abnormal leaf hairs that are forming on the undersides of these galls actually provide a nice little habitat for the mites. Russeting and deformation of fruit can also be caused by these types of mites. This is a type of Erinea gall on river birch. I don't think this is a local condition, but at least you can see what Erinea type gall would look like. And then sometimes on maples, you'll see these blister type galls also caused by an Eriophyid mite. So, moving on from the other types of symptoms, other kinds of species of Eriophyes, we'll talk a little about Rosette. Now, the mite itself, we know, is not the cause for this witch's broom condition. You can have mites on rose plants and not have the witch's broom or not have the discoloration. So we know that it's not the mite itself, but the mite is serving as a vector for this negative sense RNA virus that's causing the plant symptoms. And this was confirmed back in 2011 with some good DNA type studies that confirm this. But when going back in the literature, I found that it was actually back in 1968 that the first paper came out that demonstrated the transmission of this pathogen by the Eriophyid mite. And they looked to see, first of all, if they could transmit the disease via mechanical transmission. They were able to transmit the disease with grafting, but they were not able to transmit it with just the spider mites as well. You might think, well, what about spider mites? Could they transmit this disease? They could not get the disease transmitted by spider mites, but they could with this Eriophyid mite, Phyllocoptes fructiphilus. So this is the species that we're concerned about with this disease. And this is just a picture of those early studies. We had a 1968 study here with Allington and another study about 10 years later. So both of the early authors were able to show that Phyllocoptes were the ones that were transmitting the disease, and they also showed that mites taken from asymptomatic plants were not capable of transferring the symptoms. So that kind of nails it down as far as the mite's concerned. It doesn't mean it's the only way the disease can be transmitted, but it does suggest very strongly that this is the primary culprit. So this is Phyllocoptes fructiphilus. It's about 140 to 170 microns long. Now, micron is one of those measurements of length that's really hard for us to conceive. But to give you a little bit of perspective, a human hair is about 50 microns in diameter. So the width of these mites is about the same as the width of typical human hair. Originally, these were discovered in wild rose hips, and the multiflora rose particularly, and that's thought to be one of its favored hosts. These mites live between the leaf petioles and the axillary buds. So you're going to see them in those types of locations. If you want to go looking for these mites, and you probably need a very good hand lens, 10 or 15x hand lens, or maybe a good microscope, cut some of these stems and bring them back and look at them under a microscope. And you should be able to find them with it may take a little time. They're not necessarily that common. I've had a hard time finding them personally, but this is where you would look. And this is what you're kind of looking for. You can see those two little yellow, I don't know if I can point this out with a pointer. No, I can't. But the two little yellow objects right here are the mites. So they're going to be very small. Okay, so the Phyllocoptes are going to be most abundant on multiflora rows. I think I said that. They're how do these things get around? And that's the big question. If I could answer that question definitively for you today, I think I probably would have done my job. I'm not sure I can answer it definitively, but I can throw out to you the ideas that are out there. First of all, if you have two plants that are touching one another, if you plant your roses close enough to where they're in physical contact, undoubtedly these mites can transfer from one plant to the other that way. Another suggested possibility is wind. I've read papers that say it's not the wind, and I've read papers that say it is. The very first paper done on this mite, they looked at it in greenhouse settings, and they found that they could have infected roses in a greenhouse and in two separate areas, and the disease never seemed to get from one set of roses to the other. And that would suggest that perhaps wind is a carrying agent, or else possibly insects that wouldn't be present in the greenhouse. So it's either wind or insects are probably, in my mind, the most likely culprits for getting these mites transferred from one to the other. And talking to Steve Cheney just a few minutes ago, he was talking about one thing he's noticed is that sometimes you can get the disease show up in an isolated rose, one that's not necessarily close to any other roses. And that might suggest more of an insect-transmitted mechanism. Also keep in mind that you could possibly transmit these mites from one plant to the other, working in roses and getting them on your clothing and moving them to another site. So that's a possibility that we can't rule out either. Okay, so this Phyllocoptes fructiphilus, what does it feed on? It turns out it survives on a wide variety of rosa species and cultivars. It'd be nice if it didn't do well on some varieties, and maybe that's something that will be discovered in this upcoming research project. But as far as we know right now, it's not very picky on what kind of rose. There's no known resistant cultivars at this time, and there's no known biological control agents. It would be great if we had some kind of biocontrol agent, a fungus or another mite or something that we could release on these plants and wipe out the Phyllocoptes mites, but we don't have anything like that at the present time. Now we do know that the mite seems to prefer the tender new growth of a plant, and that drought has negative effects on the mites. When it's really hot and dry, the mite populations go down also. Now, one of the things that's especially challenging about this mite is it reproduces extremely quickly. It only takes a week for it to go from the egg stage to an adult stage. So that's an extremely short generation time for these mites, which just should give us some respect for the potential of these mites to infest our rose plants. So, some of the challenges that we face with rose rosette. These mites are not walking around on the leaves in the open where we could maybe spray them and kill them. They're actually hiding, and they hide inside fast-growing tissue. So what that means as far as a spray type strategy would go, if you're spraying plants, well, the plants are constantly growing, right? So you have to keep spraying them in order to keep a killing residue on a plant to kill these mites. So there's a challenge when you've got mites that live in the growing points of plants. They reproduce rapidly, as we just pointed out. They're easily transferred via some mechanism to other plants, either wind or humans or insects. And there are a few good insecticides that we know of. I was scouring the internet for information, actually good information about mite control, Eriophyes mite control, and I was having a hard time finding anything on this. There are a few publications, extension publications, that offer suggestions, and these are some of the suggestions that I was found, but I couldn't find any definitive research that had been done on these, so take all of these with a little bit of a grain of salt. Horticultural oil is probably the greenest product on this list. It should not be that dangerous to beneficial insects or pollinators or anything like that. Abamectin is a semi-systemic insecticide. It's a good miticide that can be applied to plants and will penetrate tissues, and that would be good for Eriophyes mite. That's probably would be my number one recommendation if you were using an insecticide. Bifenthrin is a very commonly found insecticide. You can see Orthomax is an example of that. That is one of the few pyrethroid insecticides that is also a miticide. I don't know if anybody's done any real research on this. It's just one of the products that are listed. Apparently, one of the insecticides that has been tested and works is Sevin, which is an old standard from many years back. And surprisingly, it's not systemic, but surprisingly, it seems to have some impact on these mites. And almost everyone will tell you that you need to apply these insecticides on a bi-weekly basis to keep the mite populations down. Now, I'm thinking, when I hear that kind of recommendation, if I'm growing an earth-kind rose, or a rose that I'm planting specifically because it's hardy in the environment, and I have to spray it every 2 weeks all summer long, I'm not going to do that. So insect control in general, or mite control in general, is not a real effective or practical way to control these mites. So there's really no practical or proven pesticide options for these mites at this time. I'm sorry to give you the bad news. I wish I could be the bright light in the seminar today, but I'm not going to be, unfortunately. So what are some IPM steps, integrated pest management steps, that we might be thinking about with regards to these mites and this disease? Greg's already alluded to this. Certainly eliminating nearby roses, especially multiflora roses. If you've got wild roses that are multiflora type, boy, you don't want those anywhere near your roses. Get rid of them. They're not going to make anybody cry if you pull out the multiflora roses, so should be no guilt there. Space your plants so they don't touch. I know this is not an architectural recommendation, landscape architect recommendation. We like to see nice beds of plants that are contiguous. And I don't know what to tell you about that, except for if you're just concerned with controlling the mites, spacing is going to help probably to some extent. Destroy your infected plants, as Greg's already mentioned. Consider spraying uninfected plants next to a removed plant. Now, this is the one thing, one possible insecticidal approach that has some possible merit when it comes to this mite. If I found a single rose in my garden bed, and it was the only plant in my garden that showed any symptoms, I would consider treating the roses around that area for a few months and hoping that maybe I got by and the disease had not spread to adjacent plants. And this is just another recommendation that's put out by some extension publications. Heavy pruning of plants in the winter followed by a horticultural oil spray, presumably the oil spray being to try to kill some mites that might be in some of the residual buds after you prune the rose. And that's about all I have to say about the mites. Are there any questions? Yes?
Does 7 hurt your other beneficials?
Yeah, so unfortunately, a lot of these insecticides do have negatives on beneficial insects. And 7 is a pretty broad spectrum insecticide. You would want, and I should say this, that if you do decide to treat some of the roses in your garden, perhaps around a formerly infected plant, read the labels very carefully when it comes to pollinators, because you do not want to be messing up your honeybee populations or other natural pollinators. And some of these insecticides will have restrictions, like do not apply to blooming plants, in which case if you were to do that, you'd probably want to prune all the blooms off the plant prior to spraying those plants. So keeping in mind the beneficial insects and possible negative effects is important on these. Other questions?
Is cold affected?
Cold apparently does not. A lot of the studies with these mites have been done in much colder climates than where we are. The initial study was up in Nebraska, and I believe they get a few degrees colder than we do. So waiting for that cold day it's going to be a long wait, I think, to kill the mites.
Is the disease transmitted between the parent and the offspring of the Eriophyid mites? Do we know about that one?
Well, that's a sophisticated question. She asked, can the mites transovarially transmit the disease? In other words, if a female mite has the disease, will she pass it on to her offspring? As far as I know, that research has not been done. So I don't have an answer for you on that. But it's a great question. Okay.